So first, I would like to thank the IAR for the organization and giving us the opportunity to present this webinar. Let me also say a special thanks to all the participants today. Thank you for your time, and we hope this presentation will be as much as relevant for you. And once again, don't hesitate to write any questions you may have here. Today, we will talk about industrial purification by chromatography. We will follow the road of a purification development starting from lab to the industrial life of the process, passing by process calculations and engineering study with one goal, succeed to de-risk the scale-up by ensuring the process design. Many questions are coming with an industrial development. How much are we confident in the process? How much secure is the scale-up? Or which parameters must be improved and validated? All these questions could be summarized in one key question: how to de-risk your process scale-up. Maybe you have already guessed our answer, but this is done by a step-by-step approach to be able to validate key points and then to ensure each following gates. By following this approach, we better control development time and so participate to shorten time to market. Besides, we think that with a proper R&D and process development of this work, we have the possibility to work on the efficiency of the process. This is what we want to illustrate with this presentation by exploring the process of a chromatography project. At the beginning of this journey, we bring a feed to purify, followed by a bunch of questions, information to collect, and parameters to be defined. The objective is to go step by step until the industrial process with a checked list of the information. To illustrate the process, we will take the example of an historical chromatography process for Novacep, the glucose-fructose separation. This process allows to obtain an extract enriched in fructose with a range of purity and recovery. Today, we will talk about a very high purity product compared to historical process. It's an extract of 98% purity in fructose. Before to start this story, I will take a few minutes to present Novacep and why we choose to highlight the chromatography technology during this webinar. Here is a quick reminder of who Novacep is. So Novacep Process Solutions is part of Novacep Group. We are a provider of services, equipment, and process lines in purification technologies. Our projects are in the area of life science, food, and chemicals. With 130 staff people in different countries, we can provide a customized response to different purification projects in 4 main fields. First, in food ingredients with sugar, starch derivative, or dairy ingredients. We have also functional ingredients like oligosaccharides and plant proteins, for example. We are also involved in fermentation and bio-based market with amino acid purification or organic acids. And we are now involved in industrial products development with the purification of specialty chemicals and phosphoric acids, for example. Research and development teams are located in Lyon and Shanghai. Then sales and process development are internationally present in North America, Brazil, Tunisia, Thailand, Russia, and India. We reach an average of 32 industrial process per year, plus revamping work and some pilot unit installations. We have strong references in filtration and resin purification. Our 3 main technologies portfolio are Keracep for ceramic membrane filtration, Aplexion ion exchange, it could be a batch or a continuous process in ion exchange. And the third, the target of today, the Aplexion SC, meaning sequential chromatography. Hector will explain what is sequential chromatography stand for in a few minutes. To provide an entire range of services in full production line, We add technology support for upstream process, such as catalysis and enzyme, and also for downstream process, such as filtration and evaporation, for example. Here is an explanation of why we choose to present chromatography today. Over the years, we found more and more applications for chromatographic processes, and not only on pharmaceutical grade. It is an expanding area because of four main advantages: the efficiency, the productivity, the cost effectiveness, and the environmental aspect. Indeed, it's an efficient process that leads to high purity fractions with only valuable products out. It has a very low operating cost because of relatively low water usage and the possibility to recycling the condensates. On the side of the productivity, it is interesting because it's a continuous process operating 24 hours per day and 7 days a week during the year. From an environmental aspect, it's cleaner compared to other separations because it's chemical-free, effluent-free, and permit to limit energy consumption. We here illustrate the process with glucose-fructose separation, but keep in mind that it's operating in various purifications like oligosaccharides, organic acids like citric acid, for example, amino acids, and even biomass sugar on C5 or C6 sugars. So our added value is our capability to start a project from an early phase and bring it until the process delivery, and even more a step further. This is what we will talk about in the next slides of this presentation, illustrated by the example of fructose purification. The step-by-step approach I talked about previously can be explained in 8 steps. We will explain in a first time the objectives, the content, the experiments of each steps, and then in a second time, we will apply it to our case study of fructose. It starts with the first 3 steps of lab development with the project brief and the definition of feed material. Then it's the lab screening of the resin. And the last step of this trio is the in-house numerical process simulation. Then comes here again a trio to ensure the process design with a first and important breaking point, the first process evaluation. Then we go to the pilot scale validation before to go to the process design. Next, the more industrial steps with engineering and commissioning to deliver the application SC system. An important point is not to stop here, but go further with performance management to ensure the life of the equipment and performances. I will now turn it to Hector to start with a short presentation of chromatography.
So now, let's remind the power of chromatography with the different modes to raise separation challenges in many fields, from petrochemicals and food industry to pharma and biopharma. Amongst the different chromatography modes, we focus in these 4 modes that Novacep currently implements at industrial scale for food and functional ingredients applications. We have the ion exclusion chromatography, adsorption chromatography, acid retardation chromatography, and size exclusion chromatography. The principle of ion exclusion chromatography is the charge of the molecule. For adsorption chromatography is specific interaction with the resin. For the acid retardation is specific interaction and difference of ionization. And for the size exclusion, it's a question of size of the molecule to purify. This ion exclusion chromatography is applied for neutral molecule and salt separation, sugars from biomass, and molasses desugarization. And target molecules, you can see is glycine, methionine, or sorbitol derivatives. In the case of adsorption chromatography, as example we're going to talk about, is glucose-fructose separation, also amino acid purification, and you have this kind of molecules that are purified by this method. In the case of acid retardation chromatography, we separate organic acids from other organics and salts, citric acid, lactic acid, gluconic or saccharide acid, and also mono and diacid separation. In the case of size exclusion chromatography, we can separate oligomers, mono D3-glycerol, and oligosaccharides like fructo oligosaccharides and galacto oligosaccharides. Those.
So just to start with the process development and the first stage, which is the project brief, we start with the definition of a feed material. So the very first point is to start with a characterization of the raw material, which comprises all necessary analytics from physical-chemical properties to very specific analysis, such as HPLC, ICP, or even biological analysis like electrophoresis or PCR, which allow to define the final quality of the product and impurities to be removed. The quantity and the nature of these impurities will determine eventual pretreatments and the strategy of purification. Second point is to consider, to keep the end in mind, that is, the purity, the recovery, and the capacity of production. By combining this information, we can establish the set of purification techniques to be sequenced in order to reach the requested specifications for the product. So now let's illustrate the approach with an example. So our example, as I can remember, is the feed. It's a high fructose syrup 42 with 42% of fructose and glucose. And fructose 98 is used in food industry, but also for other specialty or bio-based chemicals as a platform molecule. For this particular case, there is no pretreatment as suspended solids are not present and salt content is very low. Color and protein are not an issue, and hence raw material can be engaged directly into chromatography, which is the technology of choice for this enrichment of fructose. DPN is an impurity, and the term used for other saccharides present in the mixture, such as maltose. Next gate of process development is a screening of resins at lab scale. A preliminary selection of resins is done by crossing our experience regarding the application, impurities profile, and the characteristics of resins, skeleton of the beads, ligands, pore size, particle size. Pre-selected resins are tested and compared regarding the performance of the separation. Resins are selected from reputed manufacturers with the feedback of other process lines. Availability, delivery time, and quality of resins when going into industrial scale are also very important characteristics of resins proposed by Novacep. So in the frame of our practical case, a set of pulse injections was carried out on 2 resins, and we could determine which resin delivered the best separation. Separation performance is evaluated by calculating the resolution R, which combines the difference of retention times and peak width for each couple of species. For our case of HFS enrichment, the resin retained was XA1. One of the added values of Novacep for a process development is modeling and simulation, which are part of the DNA of Novacep since its creation. By combining experimental results of the current project, previous experience, and a unique in-house software tool, we increase considerably the reliability of our results and hence the confidence in estimations that we can deliver from this stage of process development. Numerical approach requires less raw material, less chemicals, and less time to explore a wider range of solutions. Moreover, it gives the capacity to test different constraints and to deduce the impact on the design of the process. And as the design has a related cost in terms of CapEx and OpEx, we access to a larger overview of economical scenario. In our practical case, we applied the protocol of experiments combining injections at different conditions, which has been refined and validated over time with the following strengths. Reduce as much as possible the experimental work. Carry out experiments in the smallest but representative lab device. Extract the information to determine parameters of a mechanistic model. This mechanistic model describes key phenomena related to thermodynamics, kinetics, and hydrodynamics. After the obtain of a model from experimental data, this model can be used to identify process settings, identify the sensitivity of feed variability and process parameters, and go into pilot scale with knowledge of parameters influence and process limitations. Now, I know you may think right now, well, simulation looks nice, it's modern, many domains of the industry use it, but for separation techniques and chromatography, I can find operating conditions just by running some tests and it will be okay, no math, no computer. Well, the point is that multi-column chromatography processes are more effective than single-column processes, but process settings are not so trivial. So just take a multi-column system and try to find process settings to meet specifications. It is like playing to see battle, okay? So, well, in fact, let's consider just 2 key process parameters of an SSMB. The zone where specifications of purity and recovery are respected has a triangular shape. You know, the blue triangle you see. And this triangle can be found faster by thermodynamics or by numerical screening. Just retain some figures. 1 cycle of an SSMB experimental can take 5 hours or 15 seconds by simulation. This means that all runs that you could do over 20 years could be calculated just in 6 days. Okay, so now that we have a tool to simulate different operating conditions, let's use each point and let's estimate CapEx and OpEx of the system. We can identify a whole zone that we can call area of performance, where we respect specifications of purity, recovery, or whatever. If we evaluate the impact of deviations, such as aging of stationary phase, variation of feed composition, and concentrations, we could find another area where performance would be assured and secured in case of deviations.
We are now at a strategic step, the first process evaluation. The goal is to build a design based on previous lab results, that means the design obtained by simulation. After calculations, a first mass balance is established with key values, number of cells, resin quantities, flow rates, for example, and even an approximation of the composition of the fractions. The calculations are effective on the unit or an entire process line depending on the demand. Then we are able to provide important consumption such as process water, electricity, streams, and the related costs. This work, done by the process engineer and supported by technology experts, will give a first evaluation of CapEx and OpEx of the system. This step is really important for two reasons. First, it gives a first budget to investigate industrial feasibility of using chromatography technology with a minimal number of experiments and a reduced feed amount. We all know that in case of ongoing development, available feed quantity is sometimes limited. Second reason is the simulation could bring to not only one, but different possible design or parameters to reach the same objective of purity and recovery. We could have different water consumption, different number of cells, different flow rates. This step will allow to compare and select the best design and the balance between cost and efficiency. Now let's move to the next slide and our case study. So what are the information we have at the beginning of this step? We are after the trio of lab steps. So we have feed information, resin screening, and numerical simulations that have been done. So we have the customer demand, for example, dry matter to treat in ton per day, which brings us to feed flow or capacity. We have the targeted purity and recovery. We have also the full description of the feed. Then we have the parameters from the numerical study. So we have the number of cells, the resin, the temperature, some settings to use the chromatography, and the flow rates. In the case of the application SC, in this separation, the extract contains the purified fructose and the raffinate. You can see here in the process design that the raffinate can be recycling in the before the isomerization. All these steps are calculated and some evaporator can be placed in the process line. So after process calculation, we have the column diameter, the total resin volume, and the productivity of the process line. The delivery of this evaluation answer to the question of the process feasibility. Is this purification technique economically feasible, and can the targeted purity be reached? What is the approximate cost of this system? And it permits to compare the possible design to select the best one. It brings us to the next phase, the pilot scale study, with generally only one design to validate and optimize.
So after this first process evaluation based on lab work and simulation, there is a mandatory step to scale up, the pilot scale validation. Through pilot tests, key process settings corresponding to suitable performances can be confirmed or adjusted. Data for sizing can be gathered. Long residence times can be tested with eventual impact on performances. And if recycling is present, not identified impurities may arise, and then corrective actions or operations can be anticipated, tested, and implemented for the definition of the final industrial process. Piloting is not only process-oriented, but can also be product-oriented, because the customer may need some kilograms of product for market tests. So now let's come back to our practical case. After making experiments and fitting a chromatographic model, a set of process settings for one of our pilots was deduced and pilot trials started. We observed a lower purity in fructose, 97%, and we investigated impurities profile. We deduced that for the working temperature and residence time of the products in the system, an impurity was generated in the system. And then it was decided to decrease temperature enough to prevent the impurity appearance, but managing pressure drop and resin behavior.
Now we are at the process design given with process performance guarantee. You will find lots of elements from the first evaluation, but with more precise number and list. For example, a list of supplier and equipment. To ensure a safe technical proposal, we pay attention to all the pilot results because each feed is different and each impurity plays a role in the separation game. So we need to take into account the weeks of pilot results. And now we have managed to minimize scale-up risk due to the process parameter calculation, and we enter in the building process. This step is starting when the offer is accepted and process parameters secured by the previous steps. Firstly, the engineering team has to secure the project by reviewing certifications, special regulation, and sometimes seismic standards. The idea is to avoid any unidentified risk during the conception. The second stage is the kickoff meetings to clearly be lined up with team and customer in terms of deadline, process, and layout plans to avoid misunderstanding point. Then drawings are started using PID from the process team. Once drawings are verified and validated, the orders are placed. All equipments are delivered in the same NovaSep place and verified by a quality control team. Then the equipments are sent in a global expedition. The delivery starts the step of commissioning. Firstly, the delivery and installation with storage instructions and installation supervision. The second step is to power on and verify the different parts of the chromatography system. Then is the crucial part of the water test. This is with only water in the system that the equipment is tested. If something goes wrong during the installation, it should be detected at this stage. Then a major element for a successful chromatography, the resin. Dozens of cubic meters of resin are loaded onto the columns. Then, when all work well with water, the installation can be run with product. A commissioning engineer is here to follow the suite on and fine-tune the parameters to reach the performances in terms of purity, recovery, and capacity. Here you can see an example of an application system with two chromatography columns here in red. Each column is divided into two cells, also in red. So it's a chromatography with 4 cells, and the piping are here in yellow. Behind the columns are the 2 evaporators here in gray. We have passed all the 8 gates to develop a chromatography project. We have just described how to design and build the process unit with a specific method for the process design. Engineering team is working for an easy to use and robust system with respect of customer standards and NovaSep quality. But the difficulty is not to reach the performance at the beginning, then maintain the goal during the lifetime of the equipment. Some events can lead to a modification of the separation parameters, like resin aging, for example, or a feed variation. That's why we have the run and perform section. It's important to work to ensure the performance and also extend lifetime of the equipment. A third aspect is the upgrade and revamp part. A change of feed nature for a new production, for example, can lead to a revamping of the system. It's also possible to upgrade an existing process with automation, for example. And a step of lab study and simulation can permit at this step to quickly calculate the new parameters and the advantages of an upgrade. Now it's time to summarize what we have seen. So the leading trio starts with the project brief to describe the feed and the objective with a focus on a possible pretreatment. Let's say, for example, that we need to use a monovalent resin for the separation, and your feed is full of divalent cation. We know that the resin will be converted onto divalent forms, so to protect the resin and extend its lifetime, we will have a pretreatment to remove divalent before the chromatography. After that, the resin choice is secured by a lab screening based on the evaluation of the separation parameters. Hector talked about column efficiency, elution volume, and peak resolution. These experiments, called pulse tests, are useful to select the best resin but we also use it to collect relevant data to fit the numerical model and make it more precise. The third step is the in-house process simulation, in which we start by a lab experiment called an overloading to collect the data to build the separation model. Once the model is done, we can simulate a range of parameters to determine the limitations of the separation and appreciate the possible performances of the chromatography system. The collected information will deserve to do a first process evaluation to give a process feasibility. This step will permit to select the best design in terms of balance between cost and efficiency. Then the separation parameters used to calculate the feasibility, or the best one in case of multiple choice, are validated by a pilot scale study. Thus, we will have the running parameters of the chromatography. After that comes the process design with the calculation of the industrial parameters based on pilot results. That's how we secured the industrial parameters. Then comes the engineering phase with the objective of ensuring industrial safety and robustness. This step is based on regulation issues, layout, drawing, order, quality control, and delivery. Then it's time to start the installation with power tests, water running, and then fine-tuning of the parameters to reach the performances. We have seen earlier that the important is to maintain the performances by investigation, reactions, and new study in case of feed changes, for example. It could be benefit to upgrade existing process and automation to continue to be the more efficient possible. Finally, not 8, but 9 steps allow to ensure the scale-up of the chromatography process by ensuring the process design, doing a robust and quality control equipment, and the possibility to follow with performance management options. To give you an idea of a typical duration for an industrial chromatography development, for the lab part, we talk about 2 months, including the first process evaluation. Then for pilot and design, it's about 3 months in average. The engineering part comprising orders is about 10 months and commissioning about 4 months in effective time. The performance management part should never stop. Now it's time to conclude, and I would like to thank you for your attention, and we will pass to the question and answer part.
Okay, so Alexandra, Hector, thank you for this very nice presentations. We have some interesting questions here. We have a first one, which is, does chromatography can be used to purify acids such as succinic acid? I think maybe Vincent can answer.
Yeah, Hector is actually, yeah. Okay.
The answer is yes. Succinic acid has already been studied and worked on it for separation of succinic acid. For sure.
Okay. Good. There is a second question you have partially answered in your last slide. There is a question about time and volumes required for such a development study. So you have answered, I think, more on the question of timing, but can you give some examples of volumes of feed that are necessary to perform the lab and the piloting study?
Okay. At lab scale, we're talking about some liters of feed. Well, it's always depending on the concentration, for sure, that you have in your feed, because you can have some rates of concentration for the very same product. Imagine for a fermentation broth, we can have 10 times more or less, depending on your process in upstream. But we count some kind of 10, 15 liters. That's the quantity that we need for a lab scale to have a first overview, first insight of what we can do. And then when we go into pilot scale, should we do a test to validate the pilot process? For example, in an SSMB, we can be more in the order of 100, 200, up to 1 cubic meter of volume, because even if we are successful to reduce the pilot time, because we don't have to make trial and error tests because we start from a starting point which is pretty good with our numerical approach. We still have to make it run about 2-3 weeks continuously so that we can see and validate performances in real functioning.
Okay, thank you very much. There is another series of questions on what kind of separations we can do with our application SC. And it's actually twofold. One participant asked about can we separate isomers with application SC? And another one asked about can we use application SC to separate, to make ternary and quaternary separations?
Okay. Regarding the question of isomers, the answer is yes, it's possible. It depends on the similarity of those isomers because there are several kinds of isomers. If you are in the case of chiral molecules, just forget it. But if we have isomers and we are dealing with them pretty frequently, this is possible. Sure, we can have a selectivity strong enough in our stationary phases to reach a separation, a performance separation. Regarding the 3 fraction separation, I think is the second question. It means that that's the part where we are capable of adapting sequences and get out of the SSMB sequential and to find another ones. And when I say find, it means that we of course consider all the IP part, which is sensitive, but we are capable of finding, and we have some in our, I'd say, portfolio of multi-column processes, configurations which allow to get this 3 fraction separation.
Just wanted to provide also more information on Hector comment on the ternary separation. This is actually something that we also have in portfolio. One typical example is sugar recovery from molasses. So when you work in a beet sugar factory, you have molasses. And today you can recover from the molasses the sugar, which is big part of the dry matter. It's close to 60%. You separate from non-sugar, but you can also recover the betaine, which is a very interesting amino acid, and there is a market value for that. And this is something which is already implemented at industrial scale, so that kind of ternary separation in continuous mode. So of course it's something that must be developed on a case-by-case basis, but it's already proven and available at industrial scale.
Okay, thank you, Vincent, for this additional answer. There is a question on the accuracy of the simulation results and how accurate are they?
Well, with our software tool, we have a confidence interval of 95%. Okay, so it means that our experimental points, our estimated points by our model are close to 95%. So we have such an accuracy which allows us to be confident to go forward and to really be sensitive in terms of if we modify something in the process or in operating conditions, we have the good behavior. It's not only qualitatively, but also pretty quantitatively.
And if we talk about the use of simulation results, we use the simulation data to provide the first calculation of process design. And this process design is precise to around 20-25% to complete the answer.
But in terms of accuracy, well, we have metrics typical from statistics like RMSE, and that's what we evaluate for each data fitting.
Okay, thank you. We have an additional question, which is what are the main benefits of an SSMB operation system compared to SMB, and is the extra cost worth?
Okay, first, I'm not pretty sure that there's an extra cost between an SMB and SSMB first. That should be validated because I'm not pretty sure. The thing is that by principle, an SMB process requires, it's a closed system, so it's a closed loop, and it means that you have to assure all over the time that you have the same, the addition of your two of inlet flow rates has to be the same as the addition of your two outlets. It means that your feed plus your eluent must have the same flow rate as the extract and raffinate fractions when they are collected. So industrially talking, this is pretty much complex to assure that 4 pumps are running 24 hours, 7 days at these flow rates. When you are in the SSMB, the same sequence, the word sequential is not for nothing. It's just to say that it's sequenced. It's kind of splitting of the work which is done by the SMB in sequences that only require a volume to pass through the system. So it's pretty much easier to assure a volume to pass through the system, but you can even variate eventually your flow rate in real time, but you can still keep this volume which is passing through the system on all over the sequences defining the SSMB. This is pretty much easier than assuring that your pumps have always the same flow rates to assure the positioning of your fronts in your system. So there's easiness to be installed, to be designed, and to be run. There is one thing that Vincent wants to add about this. So please, Vincent.
Yeah, I can add additional information about that. At Novacep, we have started for continuous chromatography. We have started implementing SMB. That was the end of the 80s or beginning of the 90s. And that was for glucose fructose separation or molasses desugarization. We have done that in the 90s, building SMB's units. What we can tell you is that today, most of them have been easily converted. It's not a big change in terms of hardware. Most of them have been converted to SSMB. And the main interest was that with the same volume of resin, it was possible to process much more dry matter. So it was possible to increase the capacity of the plants. So what we can say that today we don't build SMB anymore. It's definitely SSMB who took over and who replaced SMB thanks to many advantages, productivity on water consumption mainly.
Very nice. We have one specific question, which is, I think, for you, Hector. It's there is a question from a participant on what kind of type of chromatography can be simulated. And the question is relating a bit more specifically to ion exchange chromatography and hydrophobic interaction chromatography.
Okay. So our tools are capable of simulating ion exchange chromatography. We have the 4 modes that I have explained are what we call chromatography. So it's, let's say, elution chromatography in general. So for the acid retardation, ion exclusion, adsorption, and the 4th one, we have models for ion exchange. We can also be predictive in this field. And for the hydrophobic interaction, we would have let's say, a model which would be done based on the adsorption chromatography model. So, well, it might work or not. It's not exactly adapted to have the specificities of hydrophobic, but I think that we can reach accuracies as the one we've seen before.
Okay. Then there has been several questions on the applications of chromatography and questions of the type, can you use your system to separate this or that? So the different examples, so obviously we will not be able to answer specifically to all questions like this, and we will come back to each of the participants that have been asking this type of questions with more specific answers. But maybe Hector or Alexandra or Vincent, you can share a bit of our experience on these different molecules. There has been questions around peptides, around proteins, around potassium, around terpenes, xanthines, carotenes.
Okay. So for each one of these mixtures, the approach I would say is pretty much the same. We should identify a main driving force to make the separation. So if it's a question of peptides or proteins, as you said, we are currently working with proteins derived from vegetables, from juices. So the principles are, first of all, what is the key unit operation? For example, let's consider ion exchange that we currently use for make selective capture or selective ion exchange for some molecules. We always have to consider as I said, beginning the physical chemical properties of the mixture. So we have to deal with suspended solids. What can be the impact of other molecules which are not interesting as salts which are inside, and that can interfere the selectivity of the molecule of interest, like these proteins which are with other stuff. So we have to deal with all that and we can do it. Regarding carotenoids and terpenes, these molecules, then in that case we should apply more processes with absorbent resins. So it means that it has to be studied case by case, the nature of the molecule, to relate it to our know-how for which resins are used. Of course, we're going to look at the state of the art, project ourselves in eventually an industrial process. So it means that we have to see the freedom to operate. So all these things are considered all together for the process development. But in principle, all molecules that have been mentioned can be dealt by us. There is no limitation, because that's mainly the interest. That's why I just shown 4 chromatography modes in our presentation. But chromatography one of the widest methods for separation with a very high efficiency if you compare with other unit operations in chemical engineering. So for sure we have the chance to work with a technology which is very, very versatile.
Okay, very good. Very good. I think we have been through most of the questions, and I think we should thank all the participants for this very nice set of questions. It means yeah, this has raised your interest, so we're quite pleased for that, and thank you. I don't know, Joan, if you want to say some additional words, or maybe make a kind of general conclusion and comment.
Yes, thank you, Thibaut. Thank you, Alexandra and Oscar. Well, it has been a very interesting and very precise and technical webinar today, almost a training course. I think all our participants were very lucky to get all the information they get today. I hope that it was interesting for everyone. Should you have any additional question, any technical needs concerning chromatography technology and how to industrialize it, don't hesitate. You have all the coordinates of Novacep to do it. Otherwise, you can pass through us and we will put you through as well. Concerning presentation and this webinar has been recorded and it will be implemented in the Novacep website, and you will be able normally to download it as you wish. Just let us few days or 1 week to make sure that everything is on the website of Novacep. For the rest, I do think that Novacep will be present at BioCat as well next year. Then you can see them on site in presence and pass them their question if you have any additional question. And I would like to thank Delphine Peyou, who is not here today. She's there listening at least. Thibaut, Alexandra, Oscar, and Vincent for their participation and very nice presentation. We are sorry again for the Hector. I think you will have to change your computer. It's not fast enough to follow the flow of internet now, and otherwise everything was perfect. Thank you very much, and have a nice day to everyone. For us in France, it's time for lunch. Then have a nice lunch for all the Europeans. For the others, have a nice day or morning, wherever you are. Thank you very much. Have a nice day. Bye-bye.
Thank you.
Thank you. Bye.
Bye-bye.